the basics. Okay. Some of the basics, and then also point to some results that he's obtained with your materials. So Luke, why don't you go ahead? Okay, you're muted, Luke.
Okay, there we go. Hopefully you can see everything here and hear me okay now. Welcome to our first joint group meeting here between the Algar, Hudson, and Grant group. Talking today about applications of mass photometry to nanoscopic particles. So we'll just go ahead and start off with fluorescence microscopy. This is kind of the standard that most people are using at this time. It's got a lot of advantages like background suppression, chemical specificity, and can use stochastic optical reconstruction methods to create super resolution images, but they have some limitations fundamentally like photo bleaching, saturation, photo blinking, and labeling. ISCAT aims to overcome some of these issues. It's a common path interference technique, which gauges the relative phase shift between the backscattered light of a microscope cover slip and a scattering object. So some of the advantages is ISCAT is label free. You get to use continuous illumination, so there's no photo blinking. You get a high temporal resolution, so frame rate that you can go as high as whatever your camera lets you do, but also no time limit for the experiment. So you can run these things for half an hour, days if you really need to. And you can measure the point spread function of diffraction limited size particles. So in the field right now, it's largely being applied to single particle tracking, assembly and disassembly of biological nano objects like proteins, lipids, peptides, and viruses, and most popularly quantitative mass photometry, mostly of unlabeled individual proteins in solution. So this is the simple setup for ISCAT. It's a back reflection inverted microscope. So you just illuminate the sample and detect the backscattered light on a camera. We use coherent length light. It's just a green laser. And if you look at our sample right here, essentially what we look at is a nanoparticle that's bound onto a cover slip. What we measure is the light that's back reflected from the cover slip water interface and the light that's scattered from a nanoparticle. And it's the combination of those two electric fields that we actually measure onto the camera. What we end up with is an interference between the two of them, and we adjust our focus to maximize the destructive interference. So what you end up seeing are these dark spots, these dark point spread functions in your image, and they appear atop of a background. So we can do some median filtering or any number of things to isolate a background. And when we flat field, we get an image that consists of both the particle and unfortunately the speckle-like pattern, which is actually irregularities in the surface roughness of the cover slip. The important thing to note here is that the signal contrast that we measure is linearly dependent on the volume of the particle or the diameter cube or the mass. What does the microscope actually look like? So this is the microscope that I built in the Grant lab here. At the heart of it, we have a sampling stage over here. And if we zoom really close into it, it's essentially just a cover slip with silicon wells. And it's about 4 microliters of liquid that you put into it containing your nanoparticles. And over here is a penny for size. So it's a little ambitious that we have 7 on here. Usually we have 4 silicon wells per sample, but it's a free country, so I'll show you 7. I'll jump right into one of our applications here. So we use this technique to mass quantitate the size of gold nanoparticles. A gold nanoparticle that lands down on a cover slip will produce a signal that looks like this. And the system in particular involves a nanoparticle ranging from 5 to 15 nanometers in size. They are citrate capped and negatively charged on the surface at neutral pH, and they bind ionically to a lysine coated cover slip, which is positively charged. So What we observe is a dynamical process. So for observing gold nanoparticles landing on the cover slip, we use an image processing technique called ratiometric imaging or differential imaging. It's a dynamical image processing technique which only reveals in the features of the image that are actually changing in time. So as gold nanoparticles land and bind onto the cover slip, they appear as diffraction limited black spots. Once they're bound, they disappear into the background because they're immobilized on the cover slip. So once they're immobilized on the cover slip, they're not moving anymore. It's not changing in the image and they disappear into the background. So essentially they get dark and dark until the point where they actually bind and then they disappear. And this is what that actually looks like here. So this is in real time what you would see in an experiment on the screen. And these are particles just coming and crashing down on the cover slip. Then they're immobilized and they disappear forever. We go through and track each and every one of them, fit a 2D Gauss into it, and log each particle in our database. And then with a full library in A, you see that the particles will land uniformly across a cover slip. In B over here, we see the cumulative number of particles over, this is about a 2-minute experiment over here. So we see on the order of 1,000 to a couple thousand particles per minute landing on the cover slip. And you'll see that the rate actually decreases because the number of particles that land on the cover slip, that rate depends on the concentration in solution. And as we pull particles out of solution onto the cover slip, that rate decreases over time. We then look at the contrast value from each particle that we log, and we can bin them into a histogram. What we see is when we have 5, 10, and 15 nanometer gold nanoparticles, the strength of the signal increases with the size of the particle. So the big particles end up dark and the smaller particles end up a little bit more light. And we can log these histograms for each of those sizes, and we see as they get heavier and heavier, they go to further left, lower contrast values. So they go lower because they get darker and darker from a value of 1, which is a gray value, which is the median of the background. Then what we can do is we can create a linear fit between the contrast that we observe for each of the sizes of nanoparticles and the contrast that we observe in ISCAT. And what we see is when we compare these to TEMs for calibration, that we get really good agreement in both the actual size of the nanoparticle and the distribution of sizes. So using these same methods, we applied our mass photometry to both the polymer dots and the quantum dots that Russ supplied for us. I'll go ahead and start with the quantum dots. So we received quantum dots with fluorescent emission wavelengths of 525, 585, and 630 nanometers. I believe that the larger the size of the quantum dot, the more red the emission becomes. And I think I was told it was somewhere in the range of like 3 to 7 nanometers in size. Unfortunately, I was only able really to see results for the 630 nanometer quantum dots. So you can see this image right here is the average image of all 9,000 that I counted over the course of 3 experiments, and it's barely visible on top of the background. 0.998 as a contrast value is actually even less than the smallest 5 nanometer gold nanoparticle that we observed in the previous study. And then looking at the 525 and 585 over the course of 3 experiments, what I found was, a much lower rate of deposition. So 53 particles over the rate of a couple minutes, as opposed to closer to 10,000. What I think is happening here is there's some aggregates in solution that we're able to see, but probably not the really smallest ones. And I think it's worth noting that I sonicated these for about 10 minutes before putting them in, but these samples are a couple months old now. So I'm not sure exactly how these stand up over time in the fridge. So that's to be determined. Moving on from the quantum dots, we have a polystyrene sample in our lab. That's a standard that we just ordered. They're 50 nanometers in size, and we end up seeing a signal that looks like this. I thought that this was important to show because this is the sample that we have in the lab that most closely correlates to the polymer dots that we were given. So what I see here is kind of bimodal distribution, a couple contrast values, 0.98 and 0.956, and quite a high deposition rate. So over the course of 30 seconds, we see 727 particles. The concentration here is about 10 to the 11 particles per milliliter. So moving on, we have 3 polymer dots that we received. The first one is this F8BT, and I think this is possibly the largest size polymer, or the largest size particles we've ever looked at under the microscope. These are absolutely huge, and we get a really wide distribution in the size of these polymer dots. This is I think there's a combination of 4 experiments over here. And the deposition rate, in all cases, we get this decaying rate of deposition as the concentration depletes from the solution and it gets deposited down onto the cover slip. Then the next 2 that we see here are the PFVA and the CNPPV. These are really interesting because they have approximately similar average sizes. So they both have a contrast value around 0.995, but I can see that the width of this distribution here for the PFVA is quite a bit larger than it is for the CNPPV. And finally here, this is just a summary of all the different polymers that we looked at, where in these images over here, it shows the average of all the different particles that were counted. So we get this high signal to noise representation. I think it's worth noting that I always present this data in these contrast numbers. And in order to have these presented in, say, size, it really depends on the scattering properties of the particle that you're looking at. So for instance, with the gold nanoparticle, we had a series of standards, so 5, 10, 15 nanometers, and we knew exactly how big they were from the TEM, and we were able to compare those sizes to the measurements that we made with ISCAT. So once you create that calibration model, you can use that to convert from a contrast that you measure on the microscope to a size. What I don't have are a series of standards for the quantum dots and for the polymers. So it's definitely something that's possible. If we have some really well-controlled standards, we could create that calibration and get an immediate feedback of going from a contrast measured in ISCAT to a real size. So then this is the whole photometry side of it, where you can use light to measure the mass of a particle. And just want to thank all our group members, in particular, those people on the microscopy project. So Eric over here and Aden and Matt in particular. Thank you.
Great, Lou, thanks. So among the projects that I'm intrigued by was when I read, let me move this screen off of here. So I'm intrigued by some of the biological applications, in particular the interaction with cells. And are you still doing that sort of work? And are there interesting challenges associated with resolving the dynamics of those kinds of interactions? And let me just preface that by saying that, you know, right now we use, I think for all of these experiments, we use the poly-D-lysine derivatized cover slip just to have a positively charged field to bind the negatively charged nanoparticles. But we could use something like fibronectin and adhere cells to it and find a cell. And if that cell is there and immobile, it becomes part of the completely invisible background, although we would know from the bright field image where it is. And so we could keep a record of exactly where it is. But then we could correlate landings with the geometry of the cell. And there would be particular fireworks if there was any cytotoxicity or cell lysis that occurred as a result of the interactions with the nanoparticles. So if any of that is interesting to you, it sure is interesting to me. So let's discuss.
Yeah, very exciting. And I'm going to kick things off. Ed, your prompt there is very timely because about 15 minutes ago, I met with Will and Donnie to discuss the results we had from this morning about the dynamics of nanoparticles entering cells. So this actually could be pretty cool. I was taking notes the whole time, and I have lots of questions, but just to answer your question generally, my group has really at the moment two different types of luminescent nanoparticles that range in size from, I would say, 40 nanometers up to microns, like really quite large particles, and have different kind of mechanisms for getting inside a cell, which we are only beginning to scratch the surface of understanding. So unsurprisingly, some of the big particles we've made that are on the order of a micron across enter cells very differently than some of the very small nanoparticles. And the different types of small nanoparticles we've made, some are specifically engineered to invade cell membranes, and some of them we think are just passively transported inside. And we don't really have any ability to observe that process and know very little about it other than what we can read in the literature and see described. So being able to take some sort of microscope-based video of that occurring is pretty interesting to me. So yeah.
Let me just add one thing that might influence your reckoning about this going forward.
Mm-hmm.
We don't need any kind of luminescence or fluorescence on these dots in order to see them. We'll see them just from the refractive index properties.
Yep.
Okay. So
yep,
having said that, there you go.
Yeah. So I guess my first kind of, well, maybe I'll be quiet for a second and just open the floor to students, my students or anybody else's students, if you wanted to ask me questions of Luke, because I have a dozen questions, but I don't need to monopolize everybody's time. So any questions from my team or Russ's team for Luke?
Don raised his hand over here.
Yeah, Luke, thanks for the presentation. I was just wondering how large the F8BT nanoparticles were. You said they were the largest ones that you had tested so far.
Yeah, that's a good question. So these particles I didn't make, so I think that would actually be best answered by somebody from Russ's group. I would estimate on the order of like 70 to 100 nanometers.
Okay, yeah, we've synthesized some nanoparticles recently that are like 200 to 300 nanometers in diameter. So I was just curious to know that. And I was also just wondering with respect to like being able to correlate size to these measurements with the contrast values. You were saying that you could like correlate TEM data to this type of experiment, but some of the nanoparticles that we have, like their true hydrodynamic radius in solution is much different from TEM as measured by other methods like NTA and DLS. So is there any other way to try to derive size from this method aside from using a secondary method like TEM, or is that really the key?
Yeah, so you need some sort of standard to actually measure this up against. So in terms of our gold nanoparticles, what we found was that the value written on the bottle is actually different than the value that we measured in TEM, varying by actually quite a bit. So in TEM, when the bottle says it's 5 nanometers in size, it was actually 5.6. For 10 nanometers, it was 9.32. So to get an accurate calibration model, we found it highly beneficial to use something like TEM than to rely on something that's written on the bottle. But you're definitely correct in that you need something to calibrate the ISCAT signal against. So that's if you want to make measurements of the absolute size of it. But since the ISCAT signal scales with the size of the particle, if you're only interested in looking at like relative differences in the sizes of your products, then that's something you could differentiate without a calibration model.
Okay, great. Thank you.
Let me add one thing to that, in that we do see, and these are not really fully characterized yet. I mean, we have a preliminary characterization of it in our JPhys Chem paper, but we do see the effects of transport on deposition kinetics. And when you talk about a TEM radius versus an effective solution radius, anything that affects the transport is going to affect the kinds of things that we measure. And one of the things that we do measure is a depletion. So if you've got a static solution and you deposit for a time, you will create a concentration gradient in the solution. And if you can gauge that concentration gradient and use that as a model in your kinetic fitting, you could get some information about the diffusion coefficients and relate that to the effective size. And you used a term for this. Don, you said it was not a TEM size, but a
Hydrodynamic radius.
Hydrodynamic radius. Yeah. So that's certainly going to be what's related to the diffusion coefficient.
Yeah. And maybe I'll pass the floor here to Rupsa, who knows about the polymer dots.
Oh, hi. Just to answer the previous question about how big the F8BT P dots usually are. They vary in size between batch to batch, but I think Leo, you were right. Never quite 100, at least not the mean size, but maybe around 70 to 90. Like, I mean, 10 nanometers is not a big deal at this scale, but yeah, around that size. But I had a question. So you mentioned that for the F8BT P dots, you see very low rate of particles hitting the surface. But I wanted to ask, is that a factor of the concentration or the big size of the particles? Because I probably don't understand enough, but I would think that it's because of a concentration effect rather than a size effect, or the rate at which they hit the surface also has to do something with the size of the mass.
Yeah, that's a great question. So the number of particles that land on the cover slip is definitely dependent on the concentration. So if your concentration's too low, you're not going to see anything. You'll have a really low rate. And if your concentration's too high, it's just going to be chaotic. So there is kind of a sweet spot in terms of the concentration that you want to use. In terms of nanomolars, we usually work somewhere between 0.1 to 5 nanomolars. In particles per milliliter, that's usually in the 10 to the 11 range. So the actual concentration of it definitely matters in terms of the total number that are counted. Something that also happens is in the ratiometric image, if you have something that is really large, this is something that will be very clearly visible in the raw image. And if you get any kind of movement in the stage itself, it creates movement artifacts. This is typically not really a huge issue when you have really small nanoparticles, but once you get up to sizes of like 50, 70 nanometers, then any small movement in the stage can create artifacts inside these images. So as the particles get larger and larger, usually you want to dial back the concentration a little bit to try and minimize the total number of scattering objects in your field of view.
Okay.
We have Will over here with his hand raised.
Yeah, thank you for this presentation. This is a really cool technique, and I'm very fascinated by what we were talking about with the possibility of doing like live cell tracking of particles. And I just want to make sure I understand correctly that basically the idea is because the cells would be more or less stationary, they would fade into the background just like the cells that land on the, or the gold nanoparticles that land on the plate. Correct? So what you would be seeing is as the particles were moving through the cell, getting taken in by whatever pathway, you would see the particle moving but you wouldn't actually see the cell until you overlaid the image.
Exactly. Yeah, that's
Okay.
right.
Okay, great. So then with that in mind, what kind of time frames can you work with for time lapsing, both in terms of like time between images and also total time?
I think that, well, I'll preface it by saying whatever you want. Limited to how resilient is your sample. So when you're looking for like really fast time scales, we record at about 1,000 frames per second.
Wow.
But in theory, you can go for hours, days, and then you run up to limits of, okay, I need a certain amount of light to be able to fill the exposure of the camera at 1,000 frames per second. It's quite a lot. So what is the photothermal stability of your biological substance? And then also, how much hard drive space do you have? You could imagine that if you're recording at 1,000 frames per second for a day, there's not a hard drive in existence that's going to be able to keep all that data. So it gives you the flexibility for whatever you want to look at. So if you want to look at something at a really fast time scale, you can definitely do that, but typically for not as long. But if you want to observe a process that's happening over days and you're okay with a low temporal resolution, then that also works too.
Right, okay, so the limiting factor is more the sample rather than the instrumentation.
Well,
right.
the camera has a maximum frame rate of 10,000 frames per second, but that would involve a certain lack of signal averaging. So you would probably, your signal would suffer at that rate, but that's absolutely the fastest you could push it.
Great, thank you. Yeah, that answers my question. Thanks so much.
All right. Seeing no other hands for the moment, I'm just going to fire off half a dozen questions if you don't mind. So before I get really technical, I guess what I would like to ask for the grant group is what sorts of questions would you be most interested in? So do you think that the publication space of this field is still at a point where papers about like the technique itself or instrument validation or whatever is exciting, or is that kind of established at this point and you're looking for applications to other disciplines and things?
I think what we want, I mean, we have to have something that distinguishes the application in the frame of what questions are being answered. I think that just demonstrating that you can see nanoparticles is not really of particular interest. I think you want to go a little bit beyond that.
Okay, good. Yeah, that's I'm trying to get a sense of what stage it's at, because there's a point where just the fact that you could do a TEM is a Nature paper.
Right.
Yeah. But then eventually what people understand what TEM is, you need to use it for things.
So before we move on from there, this right here is like 95% of the publication space for ISCAT right now. Mass photometry on proteins in solution. So this is a calibration curve for the contrast that you observe in ISCAT versus the weight of a protein. So they scale down from like 30 kilodaltons up to 800 kilodaltons. And in terms of hydrodynamic radius, that ranges from about 4 nanometers on the low end to about 12 nanometers on the high end. So by observing things that are like 50, 70 nanometers in size, we're really pushing far beyond where the field is right now in terms of the dynamic range of the instrument.
Yeah, so that's true. And also the idea that you're applying it to a complex material as opposed to a molecular protein.
Right.
So those are new aspects. And I'm not aware of many papers out there that have looked at even at polymer beads, much less these highly engineered polymer systems that you guys are dealing with, synthesized dots or.
Yeah, I've not seen anything in the literature that uses mass photometry for polymer dots or quantum dots at this stage.
Interesting.
And I can tell you so far, there have been people who've laid down synthetic membranes on a cover slip and watched the aggregation of proteins on membranes. That's an aside. In a science paper that looked at 100 other things, somebody mentioned this with I think it's a Parkinson's amyloid protein, but there is an application there that's pointing toward that no one has pursued, that's related to cells, and that's very much related to the things you see in your large polymer dots, and that is certain configurations of macromolecules are cytotoxic in very threatening ways to living systems, and so there are amyloid diseases where proteins polymerize and then through polymerization become quite pathogenic. And if you've got polymers that are doing the same thing, that also is, I think, a very interesting kind of avenue to pursue. And nobody has done this, to my knowledge, where the actual substrate for detecting this is a cell.
Mm. Okay, that is coming together for me as a pretty interesting thing to think about. So in terms of just sizing a nanoparticle, like we have lots of ways to size a nanoparticle where you could use, DLS or nanoparticle tracking analysis or TEM or so on. And so I guess my next question is, does sizing nanoparticles by this method offer any advantages, or would the objective be to do more than just size a nanoparticle and kind of get some information about how the nanoparticle interacts with whatever it's landing on?
Well,
I think, information.
Yeah, I think with size, you also have to reckon with transport properties. And so there's that aspect of it. You're sizing from solution. Well, DLS does that, but DLS is a little bit fraught because it has you have to apply it over a narrow range. And if you've got a broader distribution of particle sizes, that's problems. You don't have enough dynamic range in the methodology that you are using with your DLS to get an accurate reading over a larger range. Yeah, certainly there is an appeal to a turnkey DLS instrument, and ISCAT is not that, but I think ISCAT's a little bit more discerning.
Yeah, a couple more things on that. With the DLS in particular, you need to use an autocorrelation function to relate the size to the actual measurement that you make in the DLS instrument, and that's not a straightforward calculation. In ISCAT, you measure a signal that's directly proportional to the size. So I think that's a little bit more reliable, and of course you need the calibration model, but it's a start. In ISCAT, you have single particle tracking. So in DLS, you're really looking at a bulk kind of measurement. And the nice thing about ISCAT is that it's both temperature and viscosity independent. So you really have to control for those in DLS and NTA, because you're measuring the movement of the particles and trying to infer it based on the diffusion. And then with the single particle tracking, you can see more rare events. So for instance, let's look at something like, let's say, one of these samples over here, so the PFVA. What you'll see in these red dots here are 1 to 2 particles that come down that are actually much larger than the bulk itself. And they get swallowed by the scale of the y-axis on the histogram here. But I believe these are things that you may actually miss in NTA or DLS, and you'll get throughput of thousands of particles per minute. With some of our gold nanoparticles, we see upwards of 4,000 per minute.
So interestingly, DLS has the opposite problem to the one you think, is that you don't miss them, but the DLS signal is proportional to the size of the particle, I think to the 6th power.
Yeah, 6th power.
It is,
yeah. So this single particle will give you this signal that's as large as 1 million tiny particles, and it actually really messes up the data.
Yeah, no, that's what I was referring to with the dynamic range.
Yeah.
So if you've got a distribution of sizes, you're in trouble with DLS.
Yeah, that's exactly right. So with ISCAT in particular, you're talking about this. So that's the scattering signal. This is that S squared. And if you're looking at me scattering, that S term is dependent on the cube of the diameter. So if you have S squared, you get D to the power of 6. But the way that the background normalization works in ISCAT is you get a signal that's dependent on D to the power of 3. instead of d to the power of 6. So this allows you to have lower signal separation between particles that are much larger difference in size.
Excellent. So I'm just going to keep going until somebody stops me. So if anyone else would like to ask a question, please raise your hand. So you mentioned particle concentrations a few times in your measurements. Are those things that the system measures, so that you knew going into the measurement, like you knew the number of particles per mil of the solution that you introduced?
Yeah, so I actually don't know the exact concentrations, but essentially what I would do as an experimentalist here is I would give it a try, and if there's not enough particles, then I'm kind of out of luck. But if there's too many particles, then I would start doing dilutions. But I know for sure that like at this rate of deposition that I'm in a certain concentration range.
Well, and here's the thing. Once we really have a handle on this this transport phenomenon and the phenomenon of depletion, we can get a concentration from the deposition kinetics, because we're counting single molecules or single particles. And if we know the relationship between the signal we're seeing on the cover slip and what the local concentration is, or what the global concentration is, then we can model that. So I think in principle, the answer to your question, Zach, is yes, that we should be able to do that. So for example, in an extreme case, one could imagine using a stirred ISCAT solution, and there very definitely the rate of deposition and any fall off depletion of the bulk concentration should be able to give you a very accurate gauge, about as accurate as anything you could imagine measuring. I mean, comparable to Beer's law type of measurement.
So then I'm also wondering, we talked about calibration. So if we had a series of polymer nanoparticles that are made of various fun organic semiconductors, would we kind of calibrate it like we calibrate a GPC? Like we go to a company that sells polystyrene beads and buy a bunch of polystyrene beads of a bunch of different sizes and create some sort of calibration curve with those different sizes, and then use that to determine the size of our particles made from.
Yeah, that's an intriguing question, and to the extent that any hydrocarbon has the same polarizability, then you should see the same scattering cross section, and then it's just a matter of size or mass, since everything's about the same density. Okay. If that's really true, then you're in good shape. Now, that's certainly not true for gold nanoparticles because of the plasmon resonance, and it depends a lot on the frequency and you know what kind of an antenna it is for me or Rayleigh scattering. But generally speaking, particles of the same nanoscale produce contrasts in this roughly the same region. But we haven't really looked much outside that region, so I don't know how confident we can be of extending that beyond the sort of a 10 to 50 nanometer range.
Okay, because as a first attempt, that seems like a reasonable thing to do then, because we can buy a bunch of polystyrene beads really easily. We can measure their sizes by TEM to get a sense of how accurate the manufacturer specifications are and what the distribution of that size is. And then, I mean, like when you say has the same polarizability, polystyrene is not going to have the same polarizability as a charge transfer semiconductor, but the refractive index might not be that different. I can't really think of a better option. Because you definitely can't buy pre-sized nanoparticles of the stuff we make. So probably for a first pass, that would probably work. Okay, my next question is the image contrast? I think actually I know the answer to this now, but is the image contrast a function of just particle size or also particle composition? So I think the answer is both, right? Like if you have a
Yeah, I mean, again, comparing a gold nanoparticle with a styrene plastic particle, I think their scattering properties are a little bit different.
They're going to be different.
I don't know if we directly compared the contrast. Do you have that number on the top of your head, Luke, about what say a 10 nanometer styrene plastic particle would look like in contrast compared with a 10 nanometer gold particle? I have the feeling that the gold particle produces a stronger contrast.
Luke is already
definitely
Luke
Yeah.
showed that already with the quantum dots that are semiconductors of comparable size to the gold. They had a way weaker signal.
There you go.
And the semiconductors are intermediate in refractive index between gold and an organic material. So
Okay.
you're going to get a lot of variation with optical mass.
Yeah.
And then I think my last question for now is going to be, is there a maximum size?
It's interesting. Go ahead, Luke.
That's a good question. In theory, our field of view is in this case like 5 microns. So if you have something that's like 5 microns in size, it's going to take up the whole screen. That's going to be quite big. But our dynamic range is quite flexible. We're really pushing what we've done and what's being done in the field by going beyond like 15 nanometer proteins, going out to, it seems like it's working for things that are, the polystyrene beads of 50 nanometers and the polymer dots 70 nanometers. And I have evidence that we can see these polystyrene beads upwards of 100 and 200 nanometers in size as well.
Can you zoom out your field of view?
So we can zoom out. We can zoom out to a maximum of maybe 20 to 30 microns.
Okay.
And our frame rate comes down a lot.
Sure.
So, you have a certain bandwidth in the cable for the camera, and we're really pushing it at the field of view that we have in the frame rate that we use.
Okay. So then I'm going to leave it there. I'm pretty convinced that it's worth a trial of, so right now we're doing various types of nanoparticle imaging with a couple human cancer cell lines and various mammalian cancer cell lines, and using nanoparticles, like we said, on the range of like 40 nanometers at the low end to probably 200 or 300 nanometers at the largest. And so it sounds like it's worth a try to just plate some cells, use that as a substrate, introduce some nanoparticles and see what you see. And then depending on how interesting what you see is, we could probably think about framing a hypothesis then.
Mm-hmm.
What happens to the size sensitivity as a function of your magnification?
Nothing. We lose temporal resolution, and averaging. So a single image is typically pretty noisy, since we're looking at like total contrast of less than 1% relative to the background. So although we record at like 1,000 frames per second, our typical temporal resolution is actually on the order of like 10 to 20 hertz, because we have to average so many frames together to get enough signal to noise ratio to say something quantitative about the particles itself.
But as you go to higher magnification, do you not lose photons per pixel?
So yes, so the nice thing about ISCAT is you can increase the laser power as much as you want until your sample actually starts to degrade. So as long as we have enough laser power, then, we're not photo bleaching, there's no photo blinking or anything. It's just a continuous radiation, and you can continue to measure that.
So if you had, say, moderate contrast on a particle size and composition at your current magnification, and then you zoom out to say 20, 30 microns, which is kind of around the size of a human cell, are you going to no longer be able to see that particle?
So when I'm zooming out, I'm not changing any of the optics. To look at this 5 by 5 micron field of view, I'm using a smaller subset of the pixels on the camera. So I'm using
this is all digital zoom. This is not changing the objective.
Yeah, so I'm not changing
Okay.
the objective or anything. So we use basically the highest magnification objective you can find. So we have a 100x, 1.5, and a oil immersion objective here. And all I do to change the field of view is we have these beam deflectors that define the total field of view. And I can change the amplitude of the deflection here to illuminate a larger spot on the sample. And then if I reduce the frame rate, it gives me the bandwidth over the camera to look at more pixels on the camera.
Yeah, and you can when you increase the illumination field, you can also increase the laser power, because the amount of light that's falling on any given area, you can keep that the same, in other words.
Yeah.
So it just becomes a throughput problem. Nothing else changes. So yeah, in principle, we can get the information. A much more interesting question, I think, is how far we can go in terms of depth of field. So the coherence length of our lasers is not the limitation, but and so what one could imagine doing is looking at planes in the Z direction, and you know, if you got a cell, you say a cell, your cells are as much as 30 microns in diameter. Yeah,
Can be.
can be. And how high do they sit on a cover slip?
Depends on the cell line. Some spread out more than others.
Yeah.
On the order of a few microns up to probably 10.
10, yeah. Yeah. So it would be, so the 10 is, we're still within the coherence length of the laser. And so we could still get interference information. It's interesting, there is a GUI phase question that determines the phase of the contrast. And so the way the image appears in the field of view changes depending upon how big it is and where you're looking at it from, where the scattering is coming from. So these are all issues that, and in fact, this is starting to get into the regime where you asked, Zach, are there papers that you can still write about variations on ISCAT methodology and have it be publishable? That's the sort of thing that may well be publishable, just to say, you know, how do I solve this problem of having a wide field of view and especially a deep field of view.
Any other questions, guys?
So let me ask you, Zach, how hard is it to, does it add a lot of complexity to your problem to make your block copolymer dots fluorescent?
No, that it's an innate feature of them. So
Okay.
they're all by design, they're all going to be fluorescent in some manner.
Okay, but if you didn't need them to be fluorescent, is that liberating in any way?
I would be out of a job. Yeah, making them fluorescent is the reason for their existence.
I see. Okay.
So yeah, but so the reason they're fluorescent is, or the applications that we explore require that they be fluorescent.
Mhm.
And however, the sizing methods that we use don't generally. So we usually size them by like DLS or TEM or whatever else. So yeah, I think that anything coming out of our lab will glow in some manner. But whether that's used in the ISCAT measurement or not, it doesn't matter.
Yeah, it generally isn't, and it's an interesting question of what role that light might play. So Bruno, you have your hand up.
Yeah, I'm just curious about something. Can you tell particles that are coming into contact with the glass versus particles like leaving the glass? If they would come, do some chemistry and then leave, can you tell them apart?
Absolutely.
Yeah.
Well, as they arrive and turn black. And then as they leave, because of the nature of the ratiometric averaging, they leave as a white spot.
Okay, I see. Makes sense.
Yeah. So what you're taking 50 frames and dividing it into 50 frames and moving that frame division around. And if you've got particles in the background frame that are not in the sample frame, that will produce an inverted image. Any other thoughts, questions? So one of the things that we're planning to do is a protein problem where we're interested in the oligomerization of proteins. I mentioned that proteins become pathogentic when they aggregate. And so there's a group in Milan that studies a disease called AL amyloidosis, which is a heart disease that's caused by aggregated proteins, cytotoxic aggregated proteins. So we're interested in watching those aggregation kinetics, sampling from a surface, sampling a solution, and then ultimately looking at interactions of cells with those proteins that aggregate in solution.
Bruno, do you still have a question?
Oh, no, sorry, I just forgot to lower the hand. Sorry.
No problem.
Thanks for answering my question.
Great. So I think as a next step, maybe Luke, I'll connect you by email with some of my students, and you can follow up about a kind of some preliminary experiments that you could give a shot. I'm pretty keen just to see what you see.
Yeah.
And then based on the observations, we can think about maybe designing a project from there.
Yeah, definitely. And if you'd like to, anybody would like to come down to the lab and check out the microscope in action, I'm more than happy to give anybody a live demo. The ratiometric movies that you see are in real time on the microscope. So you see that as soon as you drop it in a solution. And for a minute-long experiment, you get results in roughly 3 minutes or so. So the feedback is quite quick.
And I'm happy for collaborators to develop the ability to do experiments on their own. I think that the system is robust enough that people can develop a skill of operating so that people can do their own experiments. So I'd love to see that kind of level of interaction. And then I hope that we learn some cell biology from you guys.
All right, awesome. If there's nothing else, thanks very much for the presentation. I'll follow up with my team shortly and be in touch.
Sounds good.
Yeah, thanks very much, everyone.
Yeah,
Okay,
thanks everybody for coming.
Have a great afternoon.
All right.